Okay, so this is not a bat virus directly jump into human, and this is a virus based on a bat virus, which is discovered by PLA and owned by them, and going through the engineering modified in the lab, and also going through the serial passage in the animals to make it focus on human. So that's why we can say it's very powerful for human being now. So what we can say is there are very solid evidence. Simply, I can tell our audience that it's like we check something based on our fingerprint, and for the virus, the fingerprint is their genome. So we can see all this kind of evidence from their genome. That's also why in the beginning of the outbreak, China government tried to postpone the publish of the genome sequence and even change it because they initially put some wrong genome sequence to NIH database. And these are the things later be reviewed by me through Mr. Luder's YouTube, and then they quickly the next day submit Bat woman Shi Zhenli's Nature paper, which was published on 3rd February about the RATG13 virus. And then the CCP controlled and influenced the whole scientific field, and WHO, these people, they start to tell people this is the real origin and mislead, drive the attention from the real origin of the virus to the RATG13 and build something based on that.
Okay, you're saying, I want to go back. You're saying they originally did not, or inadvertently, they did put up the genome sequence and then took it down, or they put up a false one, and because you called it out, they put up the real one?
This story is like this because on 12th January this year, the first time and people know this sequence because the China government and the other scientists upload the first sequence isolated from the patient in Wuhan to NIH gene bank database. And at that time, my husband, one of the established virologists in coronavirus field, and also WHO, the expert, he did the check, and then he told me that this is a wrong genome sequence. Which
okay, so hold it. So on 12 January,
yes,
you were told on 12 January, this is the wrong genome sequence.
Yes, my husband told me that because he did the research and he's also curious about the genome sequence.
He's also at the lab at Hong Kong and a very prominent virologist.
Exactly, he's in the same WHO reference lab and who is senior to me, and also the best helper for Professor Malik Paris and Liu Peng.
Did you think that was pretty shocking that when you talked to your husband and other scientists that they would not put up the correct genome sequence, that they would not give that to NIH to put it into their database?
Yeah, that's something very, surprised because we never thought the genome sequence should be wrong because it's like fingerprint. You get it, you upload it. There is nothing can be wrong like this. Especially my husband clearly told me that this is something very weird. The wrong things happen inside.
And I want to make sure that our American audience understands this. You're saying that couldn't have been a mistake.
Yeah,
that had to be
no,
done.
It cannot be.
Why could it not be a mistake? Why would it have to be done deliberately?
So for the real motive, I think you have to ask the government, those people who control this genome sequence and all the things. But for us, we can understand it as 12th January, they upload the wrong one, which include some wrong information inside. And based on that, the virologist, the scientists cannot trace it back to the Zhoushan bat virus, which I mean the backbone for the SARS-CoV-2 owned by PLA. But one day later, 13th January, please notice that day is the day that the first overseas cases happened in Thailand. So that means people from overseas can also get a virus sample. They also can isolate it, and also people will see the problem between the wrong genome sequence and the real.
You're saying
genome
they would be caught in a lie. This would be shown.
They can be. They know that because they didn't image their overseas patient come out so quickly. And there even no interval for this some mutant happen. So once the overseas scientists get the patient, they have the big chance to isolate the virus out. That's why on 14th January, we see the previous wrong genome sequence was replaced from the NIH database. That is the version 2. And they clearly mentioned that the version 1 was replaced, which is very rare happen in gene bank database. And from that day, if you compare it, you can see among all the known genome sequence in the database, and Zhoushan bat coronavirus, ZC45 and ZXC21, they are the one most close to the SARS-CoV-2.
Is the 14th January they put into the NIH, is that the correct genome sequence or still not?
That's the one kind of correct, because 3 days later, again, they uploaded the third version, which is a little bit longer than previous one. And I did check for some important protein. I have checked between the 14th one and the 17th one. I see it's kind of okay, but also at that time, I didn't finish all the comparison. So there may be some other wrong information, important things.
Are you also saying that one of the reasons this was changed was because your interviews, you started to go on Luda's show, the YouTube show, and started to talk about this? Was that part of the reason you think that they understood that they were going to be outed, so they made these changes?
No, this is before. The 17th is before I get deliver the message through Luda's show. But what happened is we did that show on 19th January, Beijing time at night.
Yeah.
And after that, Beijing time 20th January, Professor Shi Zhengli's Bad Woman's paper about RATG13 is submitted to Nature. And also, this one is published at the same day on the same topic on Nature with Professor Zhang Yongzhen's one, who was the one first published the genome sequence of SARS-CoV-2 and said this is most close to Zhoushan bat virus. And to note, Professor Zhang Yongzhen got punished by this paper, and his P3 lab was shut down.
Are you asserting that this is part of a cover-up by the CCP and in conjunction with either the reference lab in Hong Kong and others at either WHO, etc., to cover up their tracks, to cover up their actual complicity in this?
Yeah, cover up and even more than that, because for the experienced scientists and you have the basic knowledge, you come back to see this virus genome is like the fingerprint, as I said, and it's not difficult for them to realize the problem happened inside. It's basically like the virus and become the enhanced SARS virus with SARS functional part and with some other lethal part. And this thing is like the cow has a deer's head and has some rabbit's ear and also have the monkey's hand. I mean, please tell me how it comes from nature.
And that is because you're saying it's not in nature, and that is because this is trying to be was this a gain of function? In other words, were they doing experiments to try to come up with solutions for SARS and vaccines and therapeutics? Or is this a weaponization program to try to build something that could actually be used?
I'm the one who working on the vaccine development for several years, and I can tell you this is definitely not for vaccine, for solution of the SARS virus. Briefly, this is like if you want to make a toy gun for the kid and you should remove the bullet, you should remove the harmful part. But in this version, it's like you said, okay, I'm now making the toy gun and then I put some sharp knife on that and make some missile on that. And then I give the kid that this is your toy.
So you're saying that there's zero chance that this just came from nature and that this was a very well thought through program?
Even based on common sense, yes.